Remember, we talk about aerobic respiration. We start to talk about is glycolysis. So that's glycolysis. So we want to introduce you what it is. In this class, we only need to get familiar with the name. You don't need to remember. You don't need to know the chemistry. Biochemistry class, we need to know the structure. Every single one of them, you almost like recite it, but we don't have to do. So what it is, this is the glucose. So that's a sugar. It's a basic unit of the sugar. Will become glucose 6-phosphate, which means you add a phosphate there. Then going through a isomerization process, become fructose 6-phosphates, and then you add another fructose, then become fructose 1,6-biphosphate. Okay, so this is the first step, and you can see that. Now the question here is, where these phosphate comes, where they goes? They come from an energy currency, which is ATP. So here happen is ATP hydrolyzed become ADP and a phosphate is goes here. But then glucose 6-phosphate become fructose 6-phosphates. This is isomerization. So what means isomerization? The chemical compounds are same and the structure is different. Because the chemical compounds are the same, so molecular weight are the same. The only difference is the structure is different. So glucose 6-phosphates become a fructose 6-phosphates. And then fructose 6-phosphates become a fructose 1,6-biphosphate. Where is that phosphate comes, that is obviously same thing, ATP, hydrolyzed become ADP and the phosphate. So at this stage, in the very top of the slides, this doesn't add anything. Instead, they use 2 ATPs. So what we call? We call this the investment. This is like you buy the stock. You invested $10,000 or $100,000. You want the market to get better, then maybe tomorrow you can get $100,000. That's called investment. So that's the first stage. Now, when the fructose 1,6-biphosphate comes, it's going to go to is a branch reaction. So they go these two things, which is a branch reaction. So what are those chemicals for? The first one comes out is glyceraldehyde 3-phosphates. They become 1,3-biphosphoglycerate. Then become 3-phosphoglycerate. Then become 2-phosphoglycerate. And then become phosphoenolpyruvate. And finally becomes pyruvate. So with this process, now don't forget this is a chain. Okay, so there are two sides. They are exactly the same. So what's the final product of glycolysis? Is glucose becomes a pyruvate. But in this branch reaction stage, they generate ATP instead of they hydrolyze ATP. So what happened? You can look at here. Glyceroldehyde 3-phosphate become 1-phosphate, 1,3-biphosphoglycerate right there. They happen is NAD reduced to NADH2. And then 1,3-biphosphoglycerate becomes 3-phosphoglycerate. Where is that? Here you see 1,3-biphosphate. This is only 3-phosphate. Where is that phosphate goes? Obviously is ADP with ATP become phosphate becomes ATP. 3-phosphoglycerate become 2-phosphoglycerate. This is just a phosphate position changed. Kind of like isomerization, but we don't say it. Then 2-phosphoglycerate become phosphoenolpyruvate and finally become a pyruvate. Where is this one go? We call phosphoenolpyruvate. What means enol? Do you know what means enol? Usually means loose water. That's called enol, enolization, like in the chemistry. You see the phosphoenolpyruvate become a pyruvate. Where that phosphor goes? ADP becomes ATP. So how many ATP generated during the glycolysis? 2 ATPs. But remember, it's a branch. So total how many ATPs? 4 ATPs. Now because we hydrolyzed, we invested, we used the 2 at the beginning. So if we add up them together, -2 add +4 equals 2 ATPs. So glycolysis, how many ATP generated? 2 ATPs. But be careful. I always tell my students, this means glycolysis is an isolated cycle, which means this is not a part of the whole aerobic respiration. You always keep in mind whether your background is, if we only talk about glycolysis, nothing else, 2 ATPs. If we talk about glycolysis is part of the aerobic respiration, what this means? Which means this is aerobic respiration. This is glycolysis. This is TCA cycle. If we are talking about here is part of this whole thing, then it's a different story. Where? Right here. NAD reduced to NADH2. How many ATPs generated? There is a debate about that. In the old time, we say 3 ATPs. Right now, we say 2.5. I would stick to 3 because it's easy to do the calculation. Okay? So, here, 3 ATPs generated. So the answer is very simple. How many ATPs generate if glycolysis is part of the aerobic respiration? Is part of aerobic respiration. How many? There will be 2 ATPs plus 3 ATPs multiplied by 2. This 3 ATPs is NAD reduced to NADH2. And this equals 8 ATPs. And if glycolysis is a single isolated cycle, we'll just say, or isolated branch, we can say. It is more accuracy to say than only 2 ATPs. Now there is a question. How many ATPs during fermentation? We mentioned. Only 2, is that right? And I said all the time, why only 2? That's the 2 come from glycolysis. So what this means? This basically coupled with fermentation. So these are the 2 very important conclusions we talk about. Okay? So remember AEM as part of the aerobic respiration. Okay, now, what is aerobic respiration? TCA cycle. So we're gonna jump in there. Okay, I'll remove here. We're gonna go to TCA cycle. So what is TCA cycle? That's more chemicals. People say it's more complicated. It's not really. So we have a pyruvate. Pyruvate, going through a transition step, become acetyl-CoA. Acetyl-CoA is a very long name, to be honest. We talk about acetyl-CoA is already very accurate, very concise. And this happens is NAD reduced to NADH2. Now we go through the cycle. Acetyl-CoA. So look at the TCA cycle. There is a 3 name for TCA cycle. Why we call it TCA cycle? It's tricarboxylic acid cycle. I'm gonna remove this, okay? You know already. There are 3 names for the TCA cycle. Number 1, tricarboxy acid cycle. That's where it comes from. TCA. Second, Krebs cycle. This is coming from the scientist's name. Name's Dr. Krebs. But we also call it citric acid cycle. So, if you want to remember the TCA cycle, you must know there is an important chemical here called citric acid. Or citrate, sometimes. Okay, there's citric acid. So once the citric acid comes, what we do? It depends. In the bacteria, it's go to iso-citric acid. This is in bacteria. Some of you learn biochemistry. In the animals, plants, it's not. It's isocistronic acid. So we just skip that because we'll talk about bacteria here. So citric acid becomes isocitric acid. When you see there's something like an iso, what does it mean? Isomerization, which means the same thing as before. We talked about is the chemical structure are different, but chemical compounds and the molecular weight is the same. So isocitric acid. Then goes what? A very important chemical. Alpha-ketoglutarate. This is a very magic one. This guy can go everywhere, can even relate to the lipid cycle. Okay. Then what we have? Succinyl-CoA. Let's call it succinyl-CoA. Then what we become? Succinate. Then what we have? Fumarate. Then what we have? Malate. Malic acid, you like it, it's in the beer. Then what we have? Oxaloacetate. Then finally become acetyl-CoA. So that's a whole procedure. We don't worry about these chemicals. We don't worry about these structures. We only care about where we'll generate ATP. Okay? So, we talk about aerobic respiration. Where the ATP comes. Why we call it aerobic respiration? Because they are coupled with electron transport chain. So therefore, what is electron transport? So in a certain cell machinery, like mitochondria, for animal and plants, or fungi, for bacteria in the cell wall structure, there is a very special structure called the electron transport chain. The acronym is ETC. There are certain ingredients there which is in a good arrangement. They usually started with NAD, become FAD, become FES, FMN, then go coenzyme Q, then go to cytochrome C, then go to cytochrome A, and they go to A3 and they go to oxygen. And this coupled with electron transfer. And this contributed for the ATP generated. That's the electron transport. So there are some things we want to mention just briefly here. So why the order looks like this? So how come NAD is always at the top? That's called nicotinic adenine dinucleotides. Valving adenine dinucleotides. Why the oxygen is not always at the bottom? Because we already been doing a physical-chemical reaction. We call it two-half reaction. One means two-half reaction, half of them reduction, half of them oxidized. And then we going through two-half reaction, we give them a voltage value. And we find that NAD's voltage value is very negative. And the oxygen is very positive. And the electron is always transferred from a negative value chemical to a positive value chemical. Because more negative, it is more an electron donor, and more positive, it is more electron acceptor. Now during this process, they will generate ATP. Let's say NAD reduced to NADH2. It's all the way goes here. How many ATP generated? 3 ATPs. If FAD reduced to FADH2, so it goes from here. And you see the distance between NAD to oxygen and FAD to oxygen, the former one is larger. So, electron transfer from FAD to oxygen, this is only 2 ATPs generated. So this tells us, when we talk about energy transfer during the citric acid cycle, we only care about is a place happened NAD reduced to NADH2 and FAD reduced to FADH2. So where are they? Happen right here. Reduced to NADH2. Happen right here. NAD reduced to NADH2. And happen right here. And there is also a FAD reduced to FADH2. Happen right here. But don't forget one thing. So in our class, we talk about GTP is an energy currency. But don't forget, ATP is an energy currency. But very similar to ATP, we have GTP, we have CTP, we have UTP. All those are also energy currency. So here we have GTP is energy currency. Becomes GTP. This equilibrium to ADP becomes ATP. Okay, so how many energy currency created? 3 ATP here, 3 ATP here, 3 ATP here, 1 here, 2 here, and 3 here. So how to do the calculation? 3 multiplied by how many? 1, 2, 3, 4, 4. And there is a 2 ATP here, and there is a 1 here. So how many? 15 ATPs. And don't forget pyruvate. Through the range, there are 2 cycles. So you multiply by 2 equals 30 ATPs. So how many ATPs created through citric acid cycle and the tricarboxy cycle, Krebs cycle? It's 30 ATPs. And don't forget at the top, including a transition step. Okay, we already talked about 8 ATPs going through glycolysis. And 30 ATPs going through citric acid cycle. So how many total ATPs created during aerobic respiration? 38. That is complete aerobic respiration. Now be careful. When we talk about here, this is for bacteria. How about fungi? How many? It's 36. 30 minus 2 equals 36. Why? What's the difference between bacteria and fungi? Some people say one small, one larger. Yes. What's a major difference? Bacteria is a prokaryotes. Fungi is a eukaryotes. So, which cell machinery fungi has and bacteria doesn't have? It's mitochondria. Very good. And the mitochondria is a major place for aerobic respiration. So for fungi, what are we going to do? We're going to go inside of the mitochondria. So you have to pay, is that right? You go to the Sicilian to see the basketball between WVU and the pit. You have to pay the tickets. The same thing for they use the energy. They go to the parks. They have to pay the tickets. So that's 2 ATPs. So that's 2 ATPs is like a ticket. But where are the aerobic respiration happen for bacteria? Cell membrane system. Okay, so that's a very important thing. Now, here we want to also mention 2 things. It's very interesting. So we talk about here electron transfer related to the energy generated. Okay, I'm going to remove these. So in the history, people are trying to find how ADP created ATP. So how's the ATP created? So they did a lot of research. Same as they find the DNA structure. Until one scientist comes out. His name is Peter Mitchell. In 1932, he published a paper. So what is his theory? The theory is we had an electron transport chain. The electron is transferred from donor to acceptor. The arrangement is based on to have reaction value, the voltage value of those chemicals. The more negative is on the top, more positive at the bottom. More negative indicate it's a good or good candidate for electron donor, and the more positive value indicate a chemical is a much better acceptor. What he found, the electron transfer, at the same time, we want to make sure the voltage, the balance in the cell membrane system, so he find another chemical moves out of the cell membrane system. What is that? Everybody knows that one. That's H. That's proton. So that's proton. So the proton is accumulated there. That thin layer is very magic. It's called a potential energy. Because more and more protons are created. In the cell membrane system, they have these channels we're talking about. They're going to go back. They'll not only go back. Once they go back, they trigger ATPase. They activate the ATPase. And then what happened? ADP becomes ATP. So the ATP is created. At the same time, Peter Mitchell have found in an E. coli cell, or even in a Proteus vulgaris, in those bacterial cell has a huge amount of the flagella, he find flagella rotation, which means the real movement, spin, or we say movement of bacterial cell. This whole theory gave Peter Mitchell a great honor. It's 1932, the Nobel Prize. The name called chemosmosis. Well, this is only one of the theory. Okay, this related to one terminology called oxidative phosphorylation. But I have to be cautious is that oxidative phosphorylation is not only explained by chemosmosis. Chemosmosis is one of the theory to explain oxidative phosphorylation. But no matter what type of theory it is, oxidative phosphorylation is engaged with electron transport chain. So that's aerobic respiration. The key point is oxidative phosphorylation. Then you're going to ask, How about here? There is no NAD, there is no NADH2, no FAD. How come there is ATP comes out? This comes out with another theory to generate ATP. Substrate level phosphorylation. Regarding substrate level phosphorylation, if we want to talk, we can talk about half an hour. Relate to the potential energy, kinetic energy, Gibbs principle. We're just gonna skip here, but give you some of the concise explanation. So what happened is, we have a lot of chemicals here. It's complicated, it seems. Like, look at these. So people find some of these chemicals, let's say phosphoenolpyruvate, let's say 1,3-biphosphoglycerate. Okay, let's say phosphoenolpyruvate and 1,3-biphosphoglycerate. These chemicals have a function which is transfer phosphate. And they also use a power to measure that, called transfer power. And how large it is. This is about -60. This is about -46. It's same as the voltage value. Then we give them absolutely value. This equals 60 and this equals 46. This means these type of chemical itself have a very strong potential to transfer phosphate. Why it is important? ADP with phosphate go to ATP. This chemical itself have a phosphate transfer power. It is about 35. Phosphoenolpyruvate and 1,3-biphosphoglycerate. Their phosphate transfer power is larger than ATP itself. So the reaction is going to go this way instead of go the other way. So that's why they can create ATP. So said very simple, there are some of the chemicals during the biochemical reaction have a very strong phosphate transferring power. And the power is larger than ATP itself. Therefore, they can engage with substrate level phosphorylation. So that will be a very concise and using layman language, non-technical explanation to tell you what is called substrate level phosphorylation. Okay, so that's something interesting we get to know. We learn the knowledge. And I'll use a blackboard to explain, then we always go back to see the slides. Glycolysis. How many ATP created during the glycolysis process? There's only 2. So be careful. If only 2 ATP created, what this means? This is the isolated cycle. If it's part of the aerobic respiration, there will be 8. This is explained to you, oxidative phosphorylation. There are several important items. We just mentioned the electron transport chain, ATP synthase, this is the enzyme, and the respiration. This is the electron transport chain. Same like I draw on the blackboard. Different complex, but always remember the transfer is always from an electron donor to electron acceptor. Look at the voltage value. On the top is very negative, on the bottom is very positive. This is the electron transport chain. This is chemosmosis. So it's a very good picture. Electron transfer from NAD to oxygen. At the same time, what happens is a proton will repel outside of the cell membrane system. Typically, have lots of the enzyme. Like NADH dehydrogenase. Look at the name, dehydrogenase, what this means. Hydrogen is gone, then they removed out. Ubiquinol is a quinol, that's coenzyme Q. They finally go to the oxygen. So once they accumulate there outside of the cell membrane, it's a potential energy. We gave them a specific name called proton motive force, called PMF. So proton motive force will actually driving the proton got to go back into the cell membrane system, go to the channel. The channel most of the time is composed by the protein. And it's very important that when they go back, they activate ATP synthesis. And at the same time, what happened? They coupled with rotation of the flagella. So that's called chemosmosis. That's one of the explanations for oxidative phosphorylation. And this created or named by Peter Mitchell, a great British scientist. So we skipped this one. Okay, aerobic respiration. This is just to explain to you the transition step from pyruvic acid to coenzyme Q. And this is what we talk about, is TCA cycle. So going from pyruvate, acetyl-CoA, citric acid, isocitric acid, alpha-ketoglutarate, succinyl-CoA, succinate, fumarate, malate, and oxaloacetate. So in the major class, they go through all these chemicals. We talk about all these structures, coupled with the carbon dioxide release, and the water comes back. We don't talk about here, but just this is TCA cycle. How we calculate the 38 ATPs. Don't be confusing. So what happened is here, what I talk about, NAD reduced to NADH2 have 3 ATPs. FAD reduced to FADH2 generate 2 ATPs. This is an old rule, which means they did not take consideration of the ratio of oxygen and the phosphate. In your new textbook, not really new, compared to me, okay, compare when I was a student, they take some consideration of oxygen and the phosphate. So, NAD reduced to NADH2 is 2.5 ATP, and FAD reduced to FADH2 is 1.5 ATPs. But do you like it? No, because the 0.5 makes the calculation very tricky. That's why in my class I always say we just do 3 and 2, make it easy. But be careful, what we have here is the new calculation. They are using 2.5 and the 1.5 system. And I have another slide here to help you. This is the old one. Okay, that's the 38 ATPs. So that's what we have. This will be a very important slide, can be used in the exam because it tells you the yield, the energy yield during the aerobic respiration. Okay, anaerobic respiration. So we go back to review. What is aerobic respiration? An electron donor is an organic chemical. And the electron acceptor is an inorganic chemical. But the inorganic chemical is not oxygen, it's others. For example, it could be nitrite, it could be sulfide, it could be ferric, it could be succinate. All these are environmental microorganisms because their living environment is not that perfect to get oxygen. That's why they develop those type of skills to using other chemicals to generate ATP. Okay, now the question will be, how many ATP is generated through anaerobic respiration? The answer is less than 38. The best answer, and you can see here, is 36 or less. Now why? For those type of the microorganisms, they also have an electron transport chain. But it's modified. What means modified? The voltage distance compared to NAD to oxygen is less. That's the reason their energy is not going to be that much, because the voltage distance between the donor and acceptor is less. You can easily understand it because their donor is not that rich. That's why their ATP generated is less. But here we can also answer a question. Why fermentation only 2 ATPs? Because the fermentation only generates ATP through glycolysis in the absence of oxygen, so only 2 ATPs. Okay, but why fermentation can survive? Because there is a recycled electron carrier, which is NAD+. So that's something is very interesting. We can put them together. That will be very well to understand. Okay? I know it's a little confusing. Thursday, before we talk about poultry processing, we will do a very quick review. I was spending 10 minutes to review what we talked about so far. Okay, we're gonna do another way to explain to you, which means we skip all the chemicals. And then we can only talk about energy calculation. Okay? So this is what we learn. We learn these pieces using slides. We explain to you using Blackboard. We have this class to explain to you, but you may be confusing because we come from a different background. So next class, at the beginning, we go to the new material. We're going to do a review, and we'll let you understand it well. Okay? So that's today's class material. We're going to move on to a very important work of this classroom, of this class, is we talk about the presentation. So I always think everybody works hard. Some people are very quiet. Some people are very open-minded and a good speaker. And the presentation skill is important because you go to work, whatever you do, you have to explain the things very well. So that's why we want to try to do a presentation. So how to do it? We have, assuming all the students are here, you will be assigned here is have a group. We will do 2 to 3 students as a group. I don't know you know each other, you may not know each other very well, but this is the time to get to know each other. So we're going to do a group. That's the first one. Then once you do a group, you guys will spending about a month to do a literature review. You only need to do 1 to 2 peer review publication. And then give 15-20 minutes oral presentation, about 15-20 slides. And then at the end, write 2 to 3 pages of the summary. So very simple, like an open book exam. Okay, what are the topics? It's not limited to these, it's only an example, okay? You can do pathogen control during food processing. You can talk about a microorganism causing infectious disease, what I haven't mentioned. Or even I mentioned, you can talk more a little bit detail. You can talk about the functional beneficial bacteria during the fermented food preparation. You can talk about antibiotic resistant or rapid testing of the pathogen. And then talk a little bit about regulation, like a HACCP, like a GAP, like a FSMA. We will introduce these to you in the next couple of classes. And you also could talk about emerging pathogens. Somehow like COVID-19, it's emerging in the recent news media, okay? So these are the topics, but you can pick anything you feel comfortable or you like it. What's a presentation bodies? Include introduction, objectives, materials, methods, results, discussion, some take-home message. It's coming from the paper, These are the journals I recommend you, like Journal for the Science, for the Protection, for the Microbiology, for the Control, Applied Environmental Microbiology, LWT, Meat Science, Poultry Science, Applied Poultry Research, Dairy Science, Animal Science. You don't need to read everything. So when you read a paper, what you should do? Read the abstract first. Is that right? Then you read the introduction. Statistics analysis, I don't know whether you have in statistics class, you may not, but then you look at the results. So results could be concise, and then you look at the discussion. And here are some of the tools you can use is PubMed, the Google Scholar, Science Direct. This is coming from a big publisher called Elsevier, or the University e-library system. Okay. What is a good presentation? What is a good presenter? So I gave you example, does not mean I'm a very good presenter. So it's some different standards. First of all, you make slides. Well, my teaching style, I don't use slides very often, but I go out to give a research talk, I still using slides. The slides need